Hello everyone, I'm Yu Chen. Today I'm here to dive into the fascinating world of genetically modified organisms or GMOs. GMOs have sparked a considerable debate globally with opinions ranging from stark advocacy to vehement opposition. People are afraid of the harm of GMO, and in this presentation, we will aim to demystify GMOs, explore their applications, address common concerns, and assess their impact on agricultural health and the environment. Let's start with the basics. What exactly are GMOs? Genetically modified organisms are living organisms whose genetic materials has been altered in a way that does not occur naturally through mating or mutual recombinations. This modification is typically achieved through modern biotechnology techniques such as gene spacing or recombinant DNA technology. Then we need to figure out what is gene, and the gene serve as the genetic foundation composed of a specific sequence of nucleotides on DNA or RNA molecules, controlling biological traits and transmitting hereditary information with file basis AGTC. All genes dictate the immense diversity and the multitude of organisms on Earth, from animals, plants to microorganisms, with variation arising from differences in the quantity and the coding forms of the gene bases. Genes form the basis for the development and the application of a genetic modification technology, with modern molecular biology serving as its theoretical foundation. Gene fragments transferred through GMOs can be extracted from the targeted organism's genomes, processing specific traits or artificially to obtain desired DNA sequences. And what is organism? It means any living biological entity such as the animal, plant, or the bacteria. Okay, let's continue to dive into the question how to modify and realize organisms and genetic modification. The principle of transgenic technology involves the artificial insolution or modification of specific genes with desired genetic traits using molecular biology techniques. These genes are then introduced into the genome of target organisms through specific pathways. The expression of the introduced genes leads to heritable modification in the target organism's traits, thereby achieving the purpose of modifying biological characteristics and obtaining new organisms. This technology is referred to as transgene technology. And the common method and the tools used in transgene technology include gene guns, electroporation, microinjection, and liposons. Initially employed for studying the functionality of specific genes, transgene technology involves the introduction of foreign genes into the genomes of recipient organisms, typically model organisms such as zebrafish, followed by effective recombination by observing the traits exhibited by the recombinant organisms. The aim is to exclude the function of specific genes. And transgenic technology can be broadly categorized categorized into two types based on the approach: natural transgenism and artificial transgenism. And the transgenic technology usually used among animals involved the introduction of foreign target genes into the genome of target animals according to the proposed technique schemes. And this can be achieved by introducing the foreign target genes into recipient animal outside sperm or eggs through techniques such as cell recombination, cell fusion, genetic engineering, and chromosome engineering. Subsequently, through the reproductive techniques, this modification embryos are selected and ready to cultivate the desired transgene animals as designed. Similarly, through the live-fine bacterial transformation, we can get the process of bacterial transformation to introduce exogenous DNA into bacterial cells and express specific genes. Transformation refers to the process by which bacteria uptake exogenous DNA and integrate into their chromosome. And in this experience, a plasmid carrying the target gene PUC19 is used, and which contains antibiotic resistant genes. And back to the history, we can get a look at the development of the GMO. In 1975, Cohen successfully transferred the penicillin-resistant gene carried on the plasmid of Stachylococcus aureus into Escherichia coli, and thereby opened the curtain on the commercial application of transgene technology. And the discovery of DNA restriction enzymes by Nansen, Albert, Smith earned them the Nobel Prize in Physiology of Medicine in 1978. So let's come to the next question. Why do we use GMOs? When farmers plant their crops, they generally worry about the three things that could prevent a good yield: insects, weeds, and the weather. Most of the GMO crops growing around the world today address the problems caused by insects and weeds. Today, those who directly see the most benefit from GMOs are farmers and agriculture companies. As consumers, we probably don't perceive direct benefit to ourselves just by picking the product up off the shelf. However, with many GM crops, there are secondary benefits that shoppers are unlikely to be aware by glancing at items in the aisle, such as lower cost, less soil erosion, less pesticide application, and others. However, plants are not the only type of GMO that's used. GMOs are also used to produce many medicines and vaccines that help treat and prevent disease. Before GMOs, many common medicines had to be extracted from blood donors, animal parts, or even bacteria. And this medicine have a number of problems, including the risk of transmission of disease, inconsistent quality, or unreliable supply. And GMO medicines are more consistent and do not carry the same contamination risk. And finally, we can discuss the question: do GMOs harm health? GMOs are relatively new, and like anything new, there are conflicting viewpoints about many issues surrounding the use of this plants. And one area that draws a lot of attention is whether these GMO plants and the fruits contained them are safe to eat. And until now, there is no data to indicate the consumption of GMOs is bad for human health. How do we know? So GMOs has undergone a more detailed evaluation than any other groups of plants that we consume. In almost all cases, GMOs differ from a conventional plant by the addition of just one or two genes that produce one or two new proteins. And the origin and the function of these proteins are well understood. And the National Academy for Science, the United States #1 source for independent objective advice to the nation on matters of science and technology conclude that GMOs are safe for human health. The same conclusion has been reached by a large number of prestigious health and science organizations from around the world. In addition, over the 2 decades GMOs have been on the market. There have been no occurrences of health issues due to genetically modified organisms. As GMOs stand today, there are no health benefits to eating them over non-GMO food. However, this may change in the future as technology develop and become more sophisticated. So, thank you for your listening.